we are Group 7, and for this video, we're going to discuss our video reporting in AG 206, Lesson 5 and 6. By the way, I am Amado Richilin, and I will be going to answer and discuss the three questions, which is, what is this drug? What is the relationship of this drug to proteins? And what benefits does it give to patients who will undergo surgical operation? So without further ado, let's get started. So first question, cyclosporine and an anti-rejection drug. What is this drug? This drug is used to prevent organ rejection after a liver, kidney, or heart transplant. It is usually used in conjunction with other medication to ensure that your new organ function normally. Cyclosporine belongs to the immunosuppressant drug class. It prevents your body from rejecting a transplanted organ by slowing down your body's defense system. What is the relationship of this drug to proteins? The relationship of this drug to proteins is closely linked to binding of cyclophilin, a member of a family of high-affinity cyclosporine binding proteins widely distributed in different cell types and in different species. The cyclophilins have been shown to have peptidyl prolyl, CIS trans isomerase enzyme activity that is blocked by cyclosporine. Although this may be a factor in cyclosporine selective inhibition of cytokine gene transcription, it is still unclear whether inhibition of this activity is the mechanism through which cyclosporine exerts its effect on target cells. What benefit does it give to patients who will undergo surgical operation? The benefit of this drug to patients who will undergo surgical operation: cyclosporines weaken your body immune system to help keep it from rejecting a transplanted organ such as kidney. Organ rejection happens when the immune system treats the new organ as an invader and attack it. Cyclosporine can help treat autoimmune conditions such as RA and psoriasis. It can also help prevent rejection after solid organ transplant. Cyclosporine suppresses the immune system, which can increase people's risk of infection. It also works by suppressing the immune system to prevent the white blood cells from trying to get rid of the transplanted organ. Cyclosporine is a very strong medicine. That's all, and now let's proceed to the next reporter.
Thank you, Ms. Rachelin. Good day, everyone. I am Josephine Papa, and my task is to discuss the two questions, which is about the cyclosporine and anti-rejection drug. So, this is question number 4. How and where it was obtained? It was obtained by submerged fermentation of aerobic fungi and identified as Trichoderma falciparum, but currently identified as Tolyphocladium inflatum. Since its discovery, very few studies have been carried out to search for other microbial sources for its production. Cyclosporine, also spelled cyclosporine and cyclosporine, is a calcineurin inhibitor used as an immunosuppressant medication, and it is a natural product. It is taken by mouth or by injection into a vein for rheumatoid arthritis, psoriasis, Crohn's disease, nephrotic syndrome, and an organ transplant to prevent rejection. Questions number 5. In its cyclic peptide, how many amino acids does it have? So, 391 the cyclic peptide structures are composed of 5 to 7 amino acids and are linked to a polyunsaturated acyl chain, which can vary in length. And that's all of my task. Thank you, everyone. And now, let us go on to the next reporter.
Thank you, Miss Papa. By the way, I am Beverly Similio and I will be answering the following question first. Which among the amino acids is the key to cyclosporin's pharmacological activity? So cyclosporin is a cyclic peptide with a selective immunosuppressive action. One of its 11 amino acids is a novel amino acid, which can be written like this. The preparation of which allowed the synthesis of cyclosporin and specifically modified analogs. The second question, show the amino acid sequence within the cyclosporin ring and describe its solubility in a patient cell membrane. So this one. Cyclosporin is not very soluble in water. It is much more soluble in fats or lipids, which tells us that it will be soluble in cell membrane, phospholipids, and that it will also cross them easily and enter the cytoplasm. The best way to explain this is to look at the cyclosporin's amino acids, which can be written as this. Or it also can be written like this. Out of 11 amino acid residuos, 8 are the aliphatic amino acid leucine, valine, and alanine. BMT is derived from the non-standard amino acid, or this one. And its long hydrocarbon change means it also aliphatic. Aliphatic when applied to amino acid means fat soluble.
How is DNA replicated? Replication occurs in the 3 major steps: the opening of the double helix and separation of the DNA strands, the priming of the template strand, and the assembly of the new DNA segment. During separation, the 2 strands of DNA double helix uncoil at a specific location called the origin. Now, let's proceed to the step-by-step process of DNA replication. So the first step is the unwinding of the double helix. DNA helices is the enzymes that unwinds the DNA double helix by breaking the hydrogen bonds down the center of the strand. It begins at a site called the origin of replication, and it creates a replication fork by separating the 2 sides of DNA. The second step is the synthesis of DNA segments. So DNA replication initiates at specific points called the origins, where the DNA double helix is unwound. A short segment of RNA called a primer is then synthesized and acts as a starting point for the new DNA synthesis. An enzyme called DNA polymerase next begins replicating the DNA by matching bases to the original strand. The third step is the closing of necks. So how does DNA ligase close the neck? The ligase forms a DNA adenylate complex assisting recognition. With human DNA ligase, this forms a crystallized complex. The complex, which has a DNA adenylate intermediate, allows DNA ligase to institute a conformational change in the DNA for the isolation and subsequent repair of the DNA neck. A neck is a discontinuity in a double-stranded DNA molecule where there is no phosphodiester band between adjacent nucleotides of one strand, typically through damage or enzyme action. Necks allow DNA strands to untwist during replication, and are also thought to play a role in the DNA mismatch repair mechanisms that fix errors on both leading and lagging daughter strands.
Thank you, Miss Lini Joy. Good day, everyone. I am Marcel Velanta, and I will be the one to answer the question number 9 and 10. Okay, first, let's start for the number 9 question. Are there similarities between protein synthesis and DNA replication? For me, yes. Both protein synthesis and DNA replication occur inside the cell. Both protein synthesis and DNA replication are required for the growth, development, and functioning of organism. Number 10. How is DNA replication different from protein synthesis? The main difference between protein synthesis and DNA replication is that the protein synthesis is the production of a functional protein molecule based on the information in the genes, whereas DNA replication is the production of an exact replication of an existing DNA molecules.